All right, so we're going to talk a bit about genetics now. And we're going to start by talking about reproduction. Now, there are two different types of reproduction. First of all, what is reproduction? It's creating a new organism carrying the DNA of the parents. So creating a new organism that is carrying the DNA of the parents. And there are two different types of reproduction. There's asexual reproduction. And in asexual reproduction, this is just where one parent reproduces. So one parent produces genetically identical offspring. So genetically identical offspring. So an example for this would be bacteria. An example, bacteria, they go through something called binary fission. And in binary fission, basically what they do is they make a copy of all of their stuff, and then they divide in half. And so then they have an exact copy. And then we have sexual reproduction. And in sexual reproduction, this is where we have two parents that combine their DNA to produce a different offspring. So two parents combine their DNA to produce genetically different offspring. So what this means is they are genetically different from both parents and from any other offspring coming from those two parents. So, for example, in humans, children are genetically different from their mother, and they're genetically different from their father. They're a combination of the different genes. They're combined, and they're different. But then, if those two parents, that mother and that father, have several more children, every single child is going to be different. They're genetically different. So we go through, of course, humans go through sexual reproduction rather than asexual reproduction, but we do have something that's similar to asexual reproduction in our cells that are called somatic cells. So in asexual reproduction, or in our somatic cells, what we see is something called mitosis. And we're going to go through the details of mitosis. And so our somatic cells, soma stands for body, and these are all the body cells except for the gametes. The gametes are the sex cells. So when we're talking about sex cells, what we're talking about are the sperm and egg, or ova. So we're talking about all the body cells. We're talking about the hair cells, the skin cells, the liver cells, all of the cells in the body that are not the sperm cells and not the egg cells. So when we're talking about somatic cells, or we're talking about asexual reproduction, the cell is going to divide to produce more cells that are identical to the parent cell. So the cell divides to produce more cells that are identical to the parent cell. So again, we see this in bacteria, for example, and other organisms that go through asexual reproduction. But in humans, we don't have asexual reproduction, but we do see the same type of process happening in our somatic cells. So in this case, it's necessary. It's necessary to have identical cells. We want our skin cells to produce more skin cells. When we roughen up our skin, when we break some of our skin cells off, we fall down, we scrape our skin, we want to reproduce new ones. When our liver cells start to get old and worn out, they're not functioning as well, we want to make sure that we produce more liver cells. We don't want to produce a different type of cell, we want to produce identical cells. So this process of producing these identical cells is called mitosis. So let's see why we need mitosis. So one, we already talked about a couple here. So it's necessary. So it's necessary for growth. So going from a baby. So when a baby's born, obviously they're much, much smaller than an adult. The way that they get from baby size to adult size is through growth, and this is through going through mitosis, making identical cells. So they get bigger and bigger and bigger because they're making more and more and more of all these different types of cells in the body. We also need it for healing. So I mentioned if you fall down and you scrape your arm or scrape your knee, you're going to need to replace those cells. You've damaged the body, you're replacing the cells, and we're going through the process of healing. We want to replace those skin cells. So basically, other reasons are replacing cells in multicellular organisms. And this would be not just because of healing in the case of damaging our cells, but like I said, in old, worn-out cells. So our liver cells, over time, they've been working really hard. We have to retire them. So we break them down, use the parts, and we replace them with identical cells. Some other cases are that we have some cells that divide continually. So some cells divide continually. Not all of our cells do this. There are some. So, for example, the skin divides continuously or continually. So we're constantly replacing our skin cells. We're sloughing off our skin cells. So getting our skin cells are falling off. They're becoming dust around us all the time, and we're replacing those constantly. So they're dividing continually. Versus some cells stop dividing after they mature. So an example here would be neurons. So our brain cells, once they mature, they stop dividing. We don't go through this process of continuously making neurons like we do skin cells. Then on the other hand, or lastly, there are some cells that stop dividing temporarily, but then will divide if necessary. All right, so let's talk about this process of mitosis. So again, mitosis is happening in all of the cells except for the sex cells or the gametes. And so we're talking about the somatic cells. When we talk about the somatic cells, our somatic cells are what are called diploid. Diploid means that they have two copies of each chromosome. So what that means is we have 23 pairs of chromosome in a diploid cell. So 23 pairs giving 46 total chromosomes. What we also say when we have a diploid cell is that this is called a 2N cell. It's a 2N cell, diploid, di meaning 2, and this is because we have 2 times the number of chromosomes. So we have 2 times 23. So our total then is 2 times 23, which is our 46 total chromosomes. So we have 23 pairs, 46 total chromosomes. In this case, the reason we have 23 pairs, or 46 total chromosomes, is because we get 1 copy from each parent. So 1 copy from each parent. And this is how we're combining our parents' DNA. So we get 1 copy from mom, 1 copy from dad. That's how we get information from mom and information from dad. And then we get different alleles, and we have different information that is going to be expressed. So these copies are called homologous chromosomes. These are called homologous chromosomes. So these chromosomes are similar, meaning that they have the genes for the same proteins. But they are not identical. So let's say you get one chromosome from mom and one chromosome from dad. On these chromosomes, let's say right here, we have the gene for eye color. Now this is just an example because eye color isn't so simple as one single gene. It's just a good example because we can all see different eye colors in each other. So let's say we get one from mom, which is this red chromosome, and one from dad, which is the blue chromosome. And we're saying that this area that's right here that's in green is the coding region of the DNA in the chromosome that codes for eye color. So these are our homologous chromosomes here. So these two, this is what we're saying, homologous chromosomes. So what that means is that these two carry similar information, meaning they both contain the gene for eye color. And, of course, along here, whatever this would be, they contain a gene for this, they contain a gene up here. So they have the same genes, they code for the same things, one from mom, one from dad, but those genes are not the same. So we'll say that the one here from mom, let's say, codes for brown eye color. But the same gene over here is going to code for blue eye color. Again, it's not this simple, just an example. So they're homologous chromosomes because they carry the same information or similar information, meaning the genes for the same proteins, but the information is not identical, right? Mom's codes for eye color, but codes for brown eye color. Dad's codes for blue eye color. So these are homologous chromosomes. So our gene is eye color. And in this case, we have different alleles. And the alleles are the different variations or forms of a gene. So in this case, our two different forms of the gene are brown and blue, for example. So our gene is eye color, and the alleles are the different forms. And in this case, I said brown and blue. So mom is passing on the brown allele, dad is passing on the blue allele. So now that we have that kind of out of the way, talking about what our homologous chromosomes are, let's talk about the process of mitosis. And we're going to get into more of the homologous chromosomes and how this genetic information is passed on and what that means in the next section. So again, mitosis are somatic cells. We have 23 pairs, so we have 23 chromosomes that we get from mom and 23 chromosomes that we get from dad. And again, they're paired because they are paired as homologous chromosomes. They carry similar information. So let's take a look at mitosis. So mitosis is part of what we call the cell cycle. The cell cycle is only a very small part, mitosis, a very small part called cytokinesis. So here, mitosis. Here is cytokinesis. The rest here is what a cell is going through most of the time, which is called interphase. So this whole huge part of our pie chart is interphase. Then we have a small portion that's mitosis, and then we have an even smaller portion after mitosis called cytokinesis, and we're going to talk about each of these different parts. So the entire thing is called the cell cycle, and we're going to talk about interphase first, and then we're going to get into mitosis and then cytokinesis. So let's talk about interphase. So interphase is where a cell spends most of its time. And in interphase, when it's spending most of its time here, what it's doing is it's going through regular metabolic processes. So this is when, basically, if we're talking about a liver cell, for example, this is when a liver cell is being a liver cell. It's going through, it's detoxifying things, it's storing glycogen, it's releasing glycogen, it's going through the metabolic processes. It's just existing as a liver cell. Now then, the liver cell needs to go through the process of dividing. It needs to go through mitosis to make a copy of itself, an identical copy of itself. So in most of interphase, it's just being a liver cell. But then in part of interphase, it's actually going to switch over and it's going to go through DNA replication. And so it's going to go through DNA replication, and what that means, again, is that it's going to make copies. And what it's going to do, it's going to copy all of the chromosomes. So it copies all of the chromosomes. And this is going to happen in interphase. So a portion of interphase is where it's just being a liver cell, but then toward the end of interphase, not necessarily the end, but a part of interphase is when it's going to go through DNA replication. So it's going to make all of the copies of the chromosomes. So it's going to make identical copies of every single chromosome, all 23 pairs or 46 total chromosomes. So it's going to create 2 identical copies of each chromosome. All right, so I'm going to move this to the side a little bit here, and we're going to kind of illustrate what's happening. So again, I'm going to use the red for the chromosome that came from the mother, and we're actually going to simplify this. I'm not going to draw out 23 chromosomes, but I am going to draw out, let's say, 2. So we got 1 longer chromosome from mother, 1 longer chromosome from the father, 1 shorter chromosome from the mother, 1 shorter chromosome from the father. And then what we're going to do in interphase is we're going to make exact copies of these. When the cell goes through and it makes exact copies of these DNA replication, what it does is it keeps it attached to the original chromosome. So we make a copy and it ends up looking something like this. So that's a duplicated chromosome. Now that's also a duplicated chromosome. So for right now, these are duplicated chromosomes. So these right here, the blue ones right now, are not duplicated chromosomes. So they're single chromosomes that have not made their copy yet. But we're going to do that now. So we've made a copy, we've gone through DNA replication in interphase, and we're making copies here. So now we have our duplicated chromosomes. When our chromosomes are duplicated, we have different terms. So this side over here, that was 1 complete chromosome, and then this side over here, that was 1 complete chromosome. Now this entire thing is still called a chromosome, but the individual parts are called sister chromatids. So this is a sister chromatid over here, and this is a sister chromatid over here. So again, when we started out, we had 1 chromosome, then we duplicate it. Now in this case, we still have 1 chromosome, except now we have a sister chromatid here, and we have a sister chromatid here. So watch out for the nomenclature there, and make sure that we're sure what we're talking about. So here in interphase, now what we have are duplicated chromosomes, so all 23 are duplicated. And so now we have them kind of butterfly looking rather than our individual long chromosome. So let's move into mitosis. So that's interphase, which is a large part of the cell cycle. Now we're going to move into mitosis. The first part of mitosis, the first phase, is called prophase. Now we're going to look at all of the different phases, but for short, it's PMAT. So the first one that we're going to talk about is prophase, then we'll talk about metaphase, then anaphase, then telophase. So PMAT is the acronym you want to remember. So right now we're going to talk about prophase. All right, so there are a couple things happening at prophase. I kind of like to boil it down to 3 things. So the first thing is that the chromosomes are condensing. All right, so what this means is that in reality, what we have is our cell, right? And recall that we have a nucleus. So I'm going to drop the really so we can put our DNA in. And instead of our chromosomes looking something like this, like I just drew, what we actually have at the very, very beginning is something that's more like a big triangle. So our 23 chromosomes kind of all hanging out in this way, or 23 pairs rather, sorry, 46 total chromosomes, all kind of hanging out in the nucleus in that way. But when it's going to go through interphase and duplicate the DNA or replicate the DNA, and then when we're going to go into mitosis, in order to accurately divide the DNA up, so once we've made copies of it, in order to accurately make sure that we get, the 23 pairs of chromosomes on one side of the cell, 23 pairs of chromosomes on the other side, and make sure that they're identical 23 pairs on opposite sides to make an identical cell, we need a way to clean this up a little bit. And we do that by condensing the chromosomes. So instead of having the chromosomes hanging out kind of like yarn that's just all over the place, instead in prophase then, we condense the chromosomes, and that's when we get what I kind of drew earlier, which are the sister chromatids. And so we have, I'm going to just keep it simple here with the large pair and then the small pair. So in prophase, we have the chromosomes condensing. So our chromosomes have condensed. Remember, they're still called chromosomes, but then we have our two sister chromatids. And our sister chromatids are held together by what's called the centromere. So we have a little section here. This is called the centromere. And that's going to be important as we're moving through the stages of mitosis here. So remember, this is a sister chromatid, and this is a sister chromatid. And when I say this and this, what I'm talking about, of course, is this entire left side is one sister chromatid, and the entire right side is the other sister chromatid. So I'm going to kind of get rid of some of those words there. We'll keep centromere. And then there's, of course, a centromere in each of these that are holding the two sister chromatids together. There we go. All right. So then the second thing that's happening in prophase is that our spindle begins to form at opposite ends of the cell. So our spindle begins to form. Now the spindle is a network of microtubules. So we actually have another organelle out here called the centrosome. I'll write that up here too, from the centrosome. Not to be confused with the centromere, which we just talked about. So our centrosome is actually an organelle, and during interphase, I forgot to mention this for some reason, but in interphase we do DNA replication, right? When we talk about how we make copies of all the chromosomes. Also during interphase we make copies of the other organelles. So when we do that, one of those organelles is our centrosome, which actually looks like two kind of little gears right next to each other. So two little kind of gears. And so we have one centrosome in just a regular non-dividing cell, but then when we go through interphase, we duplicate the DNA or replicate the DNA, and then we duplicate the organelle, so we end up with two centrosomes as well. So in prophase, we have the spindle fibers begin to form. And what that means is that our centrosomes here actually start to grow these little microtubules from the centrosome. And so that's going to begin to form. And we'll see why that's important in just a moment. So it's a network of microtubules. And then the third thing that happens in prophase is that our nuclear envelope begins to break down. So the nuclear envelope begins to break down. And so what we're doing here is mitosis is the division of the DNA. So we're separating the DNA into two cells. And so in order to do that, of course, we need to move this nuclear envelope. So remember, our nuclear envelope is housing all the DNA, but how would we be able to split the DNA and end up in two different cells if we can't get it out of the nucleus? So in prophase, in the first stage of mitosis here, we start to break down that nuclear envelope. So we start to make some holes in the nuclear envelope, and it's starting to break down, so we can allow some space for these things to move around. And eventually it'll be completely broken down, but we just see the beginning of that breakdown here in prophase. So these are the three things that are occurring in prophase. So then, as we move from prophase, we're going to move into, remember, PMAT, the second stage, which is metaphase. So in metaphase, what we have is our chromosomes are going to line up in the middle of the cell. So I'm going to kind of bring this up here so we can still see prophase, but in metaphase, our chromosomes line up in the middle. of the cell. So at this point, the nucleus is completely disassembled. Oops. Disassembled. And our chromosomes are pushed into place by the spindle fibers. So then what we have here, if we draw this out, so if we look at this, the first illustration up at the top here with prophase. So at this point what we've done is we've completely gotten rid of our nuclear envelope. So at this point, as we're moving into metaphase here, we don't have any nuclear envelope anymore. And our centrosomes over here have continued to make the spindle fibers. And as they have done that, they've actually pushed themselves to opposite sides of the cell. And they have made these microtubules, the spindle fibers. And what they've done is these spindle fibers actually attach to our centromere. And they have pushed the chromosomes to the center. In the center here, where the spindle fibers have pushed the chromosomes, the duplicated chromosomes, is called the metaphase plate. So our spindle fibers are attached. There we go, at our centromere, and they've lined up with this kind of imaginary line that's down the middle. And this is called the metaphase plate. So this is the way that we can organize things, right? So if we take our duplicated chromosomes and we have them lined up one by one by one by one, with each sister chromatid facing opposite poles or opposite sides of the cell, then we have things pretty well organized. We make sure that we have all the chromosomes, they're all lined up. So all 46 will be lined up in this imaginary plane, or this imaginary line in the middle of the cell, in metaphase. And then we move into anaphase. So in anaphase, this is when we're going to pull apart those sister chromatids. We're going to pull them to opposite sides of the cell. So the sister chromatids are pulled to opposite sides of the cell by those spindle fibers. So remember, the spindle fibers are attached here at our centromeres. And you can see I have the spindle fibers on the right side and the spindle fibers on the left side here, and they're both attached to the centromere. And so then what they're going to do is these spindle fibers are going to start to pull back, start to pull them in opposite directions at the centromere. And basically what we're doing is we're ripping these chromosomes in half. So one sister chromatid is going to go in this illustration toward the left, and one sister chromatid is going to go to the right for each of these chromosomes. So this sister chromatid goes to the left, the sister chromatid goes to the right. I'm going to zoom this up a little bit so we can move into the next illustration here. So this one right here is anaphase. I can probably do a better circle than that. Alright. So then I'm just going to kind of quickly draw these organelles here. And so then what we have are spindle fibers. Our spindle fibers have grabbed onto those sister chromatids and they have ripped them apart. So now they're attached right here in the center. I'm going to fix this small one, right? The big blue one. And this one. So here we are. This is our illustration of anaphase. So, anaphase. here in anaphase, you can see that I have pulled apart the sister chromatids and are pulled toward opposite sides of the cell, so toward opposite poles by those spindle fibers. So they were attached to the centromeres, and then they ripped those chromosomes in half, taking half of the DNA toward the left side of the cell and half of the DNA toward the right side of the cell. So then after anaphase, we have what's called telophase or telophase. And telophase or telophase is kind of the opposite of prophase. So in telophase, we have the nuclear envelope and the nucleolus are going to form at each pole. So the nuclear envelope and the nucleolus form at each pole. So we, after anaphase, we have telophase, and so we then start to form the nuclear envelope over here at the left side of the cell, and form our nuclear envelope over here at the right side of the cell. So this is dotted line because it's just beginning to form at this point. So in telophase, we're starting to form that. At the same time, the chromosomes are starting to relax. Because remember, in prophase, we had a condensing of the chromosomes, so now we have the opposite, so the chromosomes are starting to relax. Our nuclear envelope is starting to build up, instead of in prophase, our nuclear envelope was starting to break down. So those things are forming at telophase, or happening at telophase. And also our spindle fibers are breaking down. Because we don't need them anymore. We've already pulled our chromosomes apart, we've pulled the sister chromatids in opposite directions, so now we don't have these anymore. The spindle fibers are gone, and now we are starting to form our nuclear envelope, and our chromosomes are starting to relax and turn back into chromatin. At the same time that telophase is happening, we actually also have cytokinesis occurring. So they kind of overlap. So telophase will start, and then as telophase is starting or going through the process of telophase, then we also have cytokinesis happening. So cytokinesis is actually the division of the cytoplasm and all of the stuff that's not the DNA. So when we talk about mitosis, mitosis is very specifically dividing up the nuclear information, so dividing up the DNA. When we talk about cytokinesis, what we're talking about is dividing the cytoplasm and the stuff that's in the cytoplasm into two different cells. So the cytoplasm is divided into two cells. And the way that this occurs is that we have what's called a contractile ring. Contractile ring. This is made of actin. And it pulls the plasma membrane into two separate cells. So it pulls the plasma membrane into two separate cells. And so then this is going to produce two 2N cells that are genetically identical. To the first cell. So if we take a look at telophase and cytokinesis, what we see here is that after anaphase is occurring, when we move into telophase, and the way that I like to think of this, if we look at this illustration here of anaphase, is that when we move into telophase and cytokinesis, in telophase we have our nuclear envelope now is forming, and then at the end of telophase has formed that contains our chromosomes, so our big red one, little red one. We have identical genetic information. So big and little red one, and then the big and little blue chromosomes. And the way that I like to think of this cytokinesis, this contractile ring, is like a belt. put around the cell. So here in the anaphase illustration, if you can imagine just putting a belt around the cell here, and then putting the belt through the little buckle area, and then having the rest coming out of the buckle. And then what you do is you just pull on this. So you're pulling on the rest of it, holding the buckle in place. And what's happening is we're just tightening and tightening and tightening and tightening this belt. And so we're squeezing in on the center of the cell. And as we're squeezing in on the center of the cell, it starts to look something like this. This spot right here, this is called invagination. So it's invaginating there. This is where we have a contractile ring of protein, so it's like a little belt that's pulling the two things together. So then as we're moving through cytokinesis, this is pulling down. This, of course, is the plasma membrane pulling in, until eventually the plasma membrane touches the plasma membrane from the other side, and then we've split the cell into two cells. Because once we touch the other side, they merge together, because all of that is fluid, and then we've split into two different cells. And that part is cytokinesis. So the telophase part is regarding the DNA. The nuclear envelope has been formed, the chromosomes are starting to relax, we no longer have the spindle fibers, and then the cytokinesis is actually dividing up the two cells, so that invagination and that contractile ring. It's going to be pulling those two cells, or pulling the one cell and splitting it into two. So then that ends our mitosis and cytokinesis. That ends the entire cell cycle. Because then after cytokinesis, then we would go back into interphase. And remember, for the majority of interphase, we're just doing regular cell metabolism. Liver cells being a liver cell, skin cells being a skin cell. And so then we have gone through this entire cell cycle here. Starting all the way back up here. Remember the cell cycle, we have interphase, and then the small portion is mitosis, which is dividing up the DNA, and then the smaller piece there is cytokinesis, which actually overlaps with mitosis, really, on the telophase part. So remember, we have interphase, and then when we move into mitosis, it's PMET. So prophase, metaphase, anaphase, telophase, and then after that we go into cytokinesis. And again, remember that when we're talking about mitosis, if we look at the illustrations here on the right, what we're talking about is making two identical cells. These are for the somatic cells, or for the body cells, and not for the reproductive cells, which are called the gametes. So in mitosis, just kind of in summary, remember, in mitosis has to do with a 2N cell, which is called a diploid cell. We mentioned that up here, a 2N cell, or a diploid cell, which means that it has two copies of each chromosome, and then we replicate the chromosomes, so we make copies of them, and then we divide it into two identical 2N cells. So we end up with two 2N cells at the end of mitosis. And if anything goes wrong with this process, anything interferes with the regulation of mitosis, it can cause tumors. So we can have uncontrolled cell division. So if they're uncontrolled, that can be cancerous, and then it can begin to spread. So mitosis is very, very important. It's important that we go through mitosis appropriately. So we go through each of these steps to make sure that we get the correct DNA in each side, and that it doesn't uncontrollably divide. It's all regulated. So now that we've talked about mitosis, we said those were just the somatic cells. Now we're going to move into our second process, which is called meiosis. Now, meiosis is part of sexual reproduction. So this is where, again, we have our two parents, combine DNA. So we're looking at the end goal here, combine DNA to make offspring that are different from each parent. So if we think about the gametes, right, which is what we're talking about here. We're talking about the gametes, also called the sex cells. These are, of course, the sperm and the ova, or the egg. These cells are not two end cells, right? Because if we are combining a sperm and an egg cell, and if both cells are 2N, so if our sperm cell was 2N and our egg cell was 2N, and we combine the two, what we would end up with is a 4N cell. So the very first cell of the baby would be a 4N cell, which means 4 copies of all of the chromosomes, which would be twice the number of chromosomes that a person needs, and that actually would not be viable. And so then, even if it were, let's say that did happen, then over here we have another couple, another 2N plus 2N making a 4N baby. Then what happens when these 4N and 4N babies come together? Then are they going to make an 8N baby? If we did this, then of course our genes would be completely out of control. Our chromosomes would be all over the place. So what we want to do is we want to maintain chromosomes. So we always want to have a 2N cell that is going to start a new human being. And then it goes through mitosis in order to make all of the other 2N cells. So what we need to do then is we need to have half of that to start with, so that when we combine the sperm and the egg, we end up with 2N. So our gametes then are going to be 1N cells. 1N cells are called haploid. So our gametes, our sex cells, our sperm, our ova, they're 1N cells, are called haploid cells, which means they just have one copy of each chromosome. So how do we get these? So how do we make the sperm, how do we make the ova so that they have half of the cell, or half of the chromosomes that every other cell in the body has? Well, this is the process of meiosis. So this is the process that produces the eggs, the ova, and the sperm. And so this is a very special process just for these cells that doesn't happen in any other cell. So similar to mitosis, we actually start out with interphase. And in interphase, this is when we have our DNA replication. So again, similar to mitosis, we have DNA replication, and so we copy all the chromosomes. And then, just like we saw in mitosis, we have identical sister chromatids held together by the centromere. So identical held together by the centromere. So this was our first stage, interphase. So we're going to copy all of the DNA, just like we saw in mitosis. We make a copy of it, that's called DNA replication. So then we have twice the number of chromosomes, but we actually don't have twice the number of chromosomes. We actually have sister chromatids that are combined at that centromere. So we have the same number of chromosomes, but now we have everything copied, and we have 2 sister chromatids per chromosome. So now that we've gone through interphase and we've made a copy of all the DNA, then we're going to move into the first stage here called prophase 1. So similar nomenclature to mitosis. We started in mitosis, it was prophase. Here in meiosis, it's prophase 1. So in prophase 1, we have the chromosomes condense. Again, because we want to move them. So we want them to be close, really tight, so that we can move them easily. Again, we have the sister chromatids are held together at that centromere. And then in this case, we have our homologous chromosomes are associated with each other. Meaning that they're going to hang out by each other. So our homologous chromosomes, rather than kind of separating out like we saw in mitosis in the metaphase part, which we'll look at in just a moment, our homologous chromosomes are going to be very, very close to each other. And this is because something special happens at prophase. What happens at prophase is that our homologous chromosomes are going to have what's called crossing over. and crossing over is a recombination of DNA. And so what happens in crossing over is that the homologous chromosomes are going to exchange parts of their DNA. And we'll take a look at that. So exchange of DNA between homologous chromosomes. So if we take a look at our cell, okay, we have our duplicated chromosomes, right? So we had our red one, our big red one, and our big blue one. Remember that's one from mom, one from dad. Okay. So these are, remember, our homologous chromosomes. So this was our individual chromosome we got from mom. Let's say this one was the individual chromosome that we got from dad. They've now duplicated, so we have the sister chromatids for each of them. But remember that these share information. Remember we talked about how we had a gene here and a gene here, and they code for, let's say, eye color. Even though dad's might code for blue and mom's might code for brown, they code for eye color. And so then that would mean that, of course, we have the exact same thing here and the exact same thing here because they're identical copies. These are homologous chromosomes. And what happens in prophase is they're going to be associated with each other. They're going to loosely be near each other. And when they are, they end up crossing over, which means that their little arms and legs end up crossing over each other, as the term implies. So we have that one is going to cross over this blue one. And then what happens is they're going to cut. So we'll cut right here and cut in the same place in the blue one, and we're actually going to flip-flop information here. So then what ends up happening is that we have our red one, and now we've got a little bit of blue because we've cut it and then we've exchanged information. And then we have our blue that now has a little bit of red on it, because they've cut and then they've flip-flopped. Same thing will happen down here with these guys, is that we'll have some crossing over. Maybe we'll have it up here on the kind of upper arms, or maybe it'll be this one with this one. Maybe they kind of fold over each other in a certain way, and then they exchange information there. And also this top one, maybe they exchange here and here, for example. And so this crossing over can happen multiple times between these homologous chromosomes, or it can happen one time, or it can happen no times. And so this is different every single time. And this is part of what gives us genetic diversity. So this is one of the reasons why, even if a mother and a father have, say, 20 kids, every single child is going to have different chromosomes, or different alleles, different forms of all of the different genes in the body, because of this crossing over. One of the reasons is because of this crossing over and this exchanging of information. So it gives us lots and lots of diversity, even between brothers and sisters. Also in prophase 1, another thing that's happening is we have our spindle beginning to form. So spindle begins to form at opposite sides of the cell, because we're going to be pulling things apart. So centromeres, we have our spindle fibers. Spindle fibers. But they're crossed over. And then 3, our nuclear envelope begins to break down. I didn't draw a nuclear envelope there, but remember there's the nuclear envelope. Alright, so that sums up our prophase 1. After prophase 1, then we're going to move into metaphase 1. In metaphase 1, we have our chromosomes are going to line up in the middle of the cell, called the metaphase plate, but with one difference. In this case, our homologous chromosomes line up at the metaphase plate. So what this means is that our homologous chromosomes, or centromere, centrosome, so we have our red one, it has a little bit of blue on it, and our blue one that has a little bit of red on it are going to be right next to each other, lining up on that metaphase plate. So remember, in mitosis, they lined up individually, but here in meiosis, let's do some crossing over on this one too here. And then so instead, now our homologous chromosomes are going to line up here at the metaphase plate. And then our spindle fibers are going to be attached at the centromeres. And that is metaphase I of meiosis. So then after we move from metaphase I, then we're going to move into anaphase I. So anaphase I. And anaphase I, similar to anaphase and mitosis. Anaphase I, we have our chromosomes are going to be pulled to opposite ends of the cell. Move this over a little bit. So if we take a look at what's happening here, instead of pulling apart the sister chromatids, instead our homologous chromosomes are pulled to opposite ends of the cell, and our sister chromatids stay together. So this entire duplicated chromosome is going to move that way. This one is going to move this way, this one, and that one. So then what we end up with is we have our duplicated red one over here, duplicated red one over here, with our crossing over. Don't forget about the crossing over that has occurred. And the blue one with our little bit of red from crossing over here. So that's anaphase I. So they've been pulled to opposite ends of the cell. So then we go to telophase I and cytokinesis. During telophase I, we have our nuclear envelope forming. Our spindle fibers break down. We don't need those anymore. And then our chromosomes are relaxing. In cytokinesis, then of course we have our invagination, remember, of the plasma membrane here. It invaginates, and eventually is going to pinch apart the cell. So then at this point what we have are two cells, and actually, in order to save here, what we have are two cells, and we have our different chromosomes here. You can see that they are now different. They're not identical chromosomes, they're not identical cells. And what we have here is we still have twice as much information as we need. See here, remember, this is duplicated information. Our sister chromatids have the same information. So if one of these cells were to be passed on, if this, let's say, was a sperm cell and this was an egg cell, and we pass these on, we would actually be passing on 2 times the information of half of the chromosomes here. So this is not done yet. We are not done with meiosis. All we have completed is the first part of meiosis, which is called Meiosis I. Now what we do, there's a little bit of a break here, and then we move into Meiosis II. And Meiosis II is very similar in nomenclature. So we have prophase II, And in prophase 2, we have all of the same things that happen in prophase 1, is where we have that nuclear envelope that was formed now breaks down. We have the spindle fibers are starting to form on opposite poles, and the chromosomes are condensed. Then we have metaphase 2. And that's where we're going to draw out metaphase 2. So first I'm going to bring this down so that we can see our chromosomes. So again, we have our chromosomes that have gone through crossing over, and in metaphase 2, instead of our homologous chromosomes lining up, because those have already been pulled apart, then now our individual chromosomes are going to be lining up at the metaphase plate, just like in mitosis. So they line up individually, like this, and like this. So we have our imaginary plates. Remember, they're held by their spindle fibers here on the centromeres. Quick here, we've got other spindle fibers, of course. And so in metaphase 2, we have them lining up in this imaginary line in the middle of the cell called the metaphase plate. And there are individual chromosomes that are lined up, not homologous pairs, because those have already been pulled apart. Then we get to our anaphase 2. And in anaphase 2, our sister chromatids are pulled apart. So just like we saw in mitosis, we're going to rip this in half, and our sister chromatids are going to go in opposite directions here. So in anaphase 2, we end up with sister chromatids being pulled apart. So if our sister chromatids are being pulled apart, if we look up here, that means that one is going to be completely red on this side, and then this one on the other side is the one that's going to have that bit of blue from crossing over. Then down here, we have one side, one sister chromatid was completely red, and then the other sister chromatid is the one that had that little bit of blue from crossing over. And then we see the same thing happening over here. So we pulled one sister chromatid to one side, and then we pulled the other sister chromatid to the other side. Now, the way that I've drawn them here, right, I kept the crossing over, the ones that had crossing over, kind of right next to each other. But when we are in metaphase 2, if we move up to this area here again, when we're in metaphase 2, these guys are going to line up independently of each other. This is what's called independent assortment. Independent assortment. What that means is that these things could have flip-flopped. So up here, if we look at the metaphase illustration, I kept the one that crossed over here. But we could have switched this. When they lined up here the second time around, the blue piece could have been on this side, and then the red piece on the other side. So it could have flipped the other way. And if it had, then these two would be switched. Same thing could have happened over here in this cell. So if we had kind of spun this guy around, and then these two would be switched, which means that this guy would be over here with this other chromosome, and then this one would be with that chromosome. So this is meaning that our chromosomes don't stay the way that they were. Independent assortment, so they sort independently of each other. That means that just because this top one up here is lined up this way doesn't mean that this one's going to be lined up this way. It can flip-flop, it can turn around, and then of course remember that we have 23 pairs, so this is going to happen with 23 duplicated chromosomes. So one will be flipped one direction, the other one will flip the other direction, and then the next time it'll flip the other way and flip the other way. So this is what's independent assortment, and this is another way that we get genetic diversity. Because each time a sperm is made and each time an egg is made, they will assort independently, and it'll be different each time. So then here we are in anaphase 2. We've pulled those chromatids apart, Then we move into telophase 2 and cytokinesis. And at this point, then we create our nuclear envelope. We break down the spindle fibers. We don't need those anymore. And then our chromosomes are starting to relax. And then we have cytokinesis. So we have that invagination that starts. That's going to divide these into 2 cells. So I will separate them into 2 cells. So then what we see is at the end of meiosis, we end up with 4 cells. Remember when we did mitosis, we ended up with 2 cells. In meiosis, we end up with 4 different cells, because notice our chromosomes are different. So our cells are non-identical cells. They only have 1 copy of each chromosome. So you can see that this one has 2 fully red chromosomes, whereas this one here has 2 chromosomes that they have the same information, but this one is partially red, partially blue, which means that this one has blue DNA at the bottom versus this one that has red DNA at the bottom. Is different. This one has blue at the top and red at the bottom. This one's completely red. So they're different information and different alleles. Same thing over here. We have red at the bottom, we have red at the top. Over here we have blue at the bottom, we have blue at the top. So every single cell is different. See, these 2 are blue chromosomes. Over here there are 2 red chromosomes. So they carry similar information because they carry the same genes, but the DNA is different. The alleles are different. So every single cell that is made is different. So in meiosis, we started with a 2N cell, right? So we started out with those 46 chromosomes, or 23 pairs of chromosomes. Then we replicated the chromosomes. So remember, we made a copy of them. So we replicated the chromosomes. After we replicated the chromosomes, remember, we had crossing over. That leads to genetic diversity. We don't see that in mitosis. And we exchange information that way. And then the cell is going to divide twice. So we start out with 1 2N cell, and we end up with 4 1N cells. And they are non-identical. And that's very, very important, because that means that whether we're talking about sperm or whether or not we're talking about egg, that every single time a sperm is released and every single time an egg is released, that they have different DNA, they have different information. That's due to this crossing over, as well as to independent assortment. That's going to give us variation, genetic variation. So then, of course, now that we have made these 4 individual cells, then we have fertilization. So some of these, if we're talking about spermatogenesis, then we have made the sperm in the male. If we're talking about oogenesis, we've made the ova, or the oocyte, in the female. And so then what we're going to do, these are the gametes. So those are the processes that make the gametes. In males, we call it spermatogenesis, which is basically going through meiosis I and meiosis II. In females, it's called oogenesis, which is going through meiosis I and then eventually meiosis II in the female. These are going to be producing our gametes, which are a 1N cell, or haploid. Remember that term as well, haploid cells. So then, fertilization is going to be a joining of the gametes to form the offspring, And of course, humans have internal fertilization, which means it happens inside the body. And then after fertilization, we have development. And development starts off with the fertilized egg is going to divide by mitosis. So the fertilized egg divides by mitosis. meiosis, which we already talked about. So the very first egg, so the sperm is going to meet up with the egg. Once the sperm meets up with the egg, that's called fertilization. So once the sperm burrows its way through the outside area of the egg and is inside the egg, the two DNA have come together. That's fertilization. When that happens, it forms what's called a zygote. So a zygote comes from fertilization, the sperm and the egg coming together and joining the DNA. Then after fertilization has happened, we have the one individual cell. The zygote is a 2N cell now because it has half the DNA from the mother, the oocyte or the egg, and half the DNA from the father, from the sperm. And now that's come together to make a 2N cell or a diploid cell called the zygote. Now that zygote, which is the fertilized egg, is going to divide by mitosis, and then it's going to grow and then mature. So lots and lots and lots of mitosis happening. So that's where we're going to end this. We talked about mitosis. You should be familiar with mitosis, the reason for mitosis, what cells go through mitosis, what the cell cycle is, what happens at each of the stages, so interphase, prophase, metaphase, anaphase, telophase, cytokinesis. And then you should be familiar with meiosis. So you should know Meiosis I, including interphase, then prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And then you should know Meiosis II. So prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. You should know that in mitosis, we start out with a diploid cell and end up with two identical diploid cells. And that's because those are our body cells making copies, identical copies. And you should know in meiosis that we start out with a 2N cell or a diploid cell, and we end up with 4 non-identical, totally different haploid cells. And these are specific to the gametes. And then when those gametes come together, that's called fertilization. It forms a zygote, and then it'll go through development, which is where that zygote then is going to divide by mitosis many, many, many times over, and eventually form the fetus, the baby that will then grow up to mature and become an adult.